Swayam Prabha. Digital India. Educated India.
Welcome you all to the next class on inorganic chemistry of life. In the previous class we have been looking at the manganese based enzymes. I said the manganese based enzymes based on one manganese, there are two based on two manganese and three manganese, and towards the end of the class we were trying to look at the corresponding reactions of the superoxide, going from the superoxide radical, superoxide dismutation of the radical to the hydrogen peroxide, and the catalase disproportionating the hydrogen peroxide to water and oxygen. And we also looking at the photosystem where the carbon dioxide is converted to the carbohydrate and the water is oxidized to oxygen. And we looked at two other reactions as well. One is the ribonucleotide reductase, where ribonucleotides, deoxyribonucleotides are synthesized from the ribonucleotides by the removal of the OH group from this 2' position and replacing it with the hydrogen. So, that is where. And one another example we have looked at was the case of the dioxygenase. I talked to you that dioxygenase will be taught to you more detailedly under copper, under iron. Okay, so let us see in this class, let us look at what is the kind of a problem that one would expect, or what kind of a damage aspects one would expect with the O2 minus superoxide radical. So, I am sure you must have, everyone must have heard something called oxidative stress. So, oxidative stress is nothing but the formation of oxygen based radicals, oxygen based anionic species, which are in turn involved in reacting with the tissue and spoiling the tissue, cell or organ, and that is what is referred as the oxidative damage. As you can see here, O2 minus dot, which is a superoxide radical, can combine with a nitrosyl group and can affect the lipids, can affect cells, can affect various kinds of things in the biological system, and all this leads to the damage of the biological tissue. So, therefore, all this is referred as reactive oxygen species. Most of the times these are referred as ROS, and some people will call this as ROS. So, I would prefer not to use the word ROS, but I would use the word ROS, reactive oxygen species. So, this reactive oxygen species is an important terminology. O2 minus dot, O22 minus, OH minus, OH minus or OH dot. So, all these kinds of things are called the reactive oxygen species, which is written as ROS. Okay. So, I will be referring to ROS at different stages. So, ROS is a reactive oxygen species which cause damage to the tissue, cells and tissue, therefore, which is referred in the form of oxidative stress. Now, let us look at the enzyme which indeed does take care of this particular O2 minus radicals and try to disproportionate these ones. So, that particular enzyme is called the superoxide dismutase. It is referred as superoxide dismutase. Since it is a manganese containing, we call it as a manganese superoxide dismutase. Of course, this is very vital in the cells because it will protect the cells from the reactive oxygen species, and thereby it will protect the cells from the apoptotic cell death. So, it will protect from that. So, that is in other words against oxidative stress. Okay. So, all these things, and this enzyme encodes the mitochondrial protein, and it forms a homotetramer and binds one manganese per subunit. There are 4 manganese for the total protein, and this protein binds the superoxide, of course, by the product of the oxidative phosphorylation, and converts the superoxide into the hydrogen peroxide, as we have seen already, and the dioxygen O2. Okay. So, that means partly it is making the harmful O2 minus to O2 which is harmless, H2O2 which is partially harmful. Okay, that we will see in a while. Let us look at the kind of reaction that happens in this particular case, and you see that the enzyme is shown over there. So, this is what one of the units of the total tetrameric protein. So, this is called the monomer of the protein. So, 4 such things will join together and form the total protein, and the 4 manganese centers, therefore, you have reactivity happening at each stage. Now, the manganese superoxide is an enzyme, and here we are showing one subunit. The manganese is surrounded by these groups, which you can see very clearly over there, one histidine, other histidine, and the other histidine oxo or hydroxo species, which is suspended or connected to the secondary force interactions coming from the protein. This is also the protein. So, this is the kind of a situation. So, what happens when you add to this enzyme 1 mole of O2 minus. So, the manganese in this state as the manganese 3 plus, and the manganese 3 plus when the O2 minus is added and the O2 minus is oxidized, means the electron should be going to the manganese. So, the manganese 3 plus, when the electron comes to the manganese center, it becomes manganese 2 plus. So, manganese 3 plus is reduced, O2 minus is oxidized, all that we would say. So, now this is O2 minus, which is oxidized O2, oxidized O2 will not stay, it will go escape away. And now, so now you have enzyme in the manganese 2 state, and this manganese 2 state enzyme in the is reactive towards the 1 more, or captures 1 more mole of O2 minus, and this will react with the manganese center and to get the oxidized and get self reduced. When the O2 minus is reduced, what will happen? O2 minus dot is reduced, it will become O2 2 minus. So, in the first stage you have O2 minus dot, and this will give away electron. Okay. So, therefore, it will give O2. In the second step, O2 minus is added, and to this you are basically taking the electron to this. So, therefore, and that will give to O2 2 minus kind of things. So, you have oxidation part, and you have a reduction part. So, this means the manganese 3 here, manganese 3 plus will go to manganese 2 plus, okay, and then manganese 2 plus will go to again manganese 3 plus. Okay. So, we will go to the manganese 3 plus. So, the manganese 2 plus will go to this. So, this O2 minus is oxidized, manganese is reduced. O2 minus is reduced, manganese is oxidized. All that you have to understand is very nicely, very simply, it is a redox reaction. Very simply, it is a redox reaction, nothing more than that, no need to worry at all. So, very simple to remember, understand the whole thing. So, first mole, the O2 minus dot gets oxidized to O2, manganese 3 gets reduced to manganese 2, and then second O2 will reduce, that means reduced form of O2 minus is O2 2 minus, which is peroxo kind of species, and this will in presence of the proton it will go as a hydrogen peroxide and go back to the manganese 3. So, the same thing is shown over there, nothing more. Manganese 3 SOD O2 minus dot plus H plus manganese 2 SOD, and this converts to water and oxygen is lost. Now, manganese 2 form of SOD with 1 more mole of the O2 minus will go to the manganese 3 O2. Okay. So, first superoxide ion is oxidized and the second superoxide ion is reduced. In the first step, the manganese oxidation state is reduced by 1, and then second state it is regained. So, I hope you understand the whole thing. Now, so in this particular enzyme, what have we created? You created or generated O2 plus H2O2. So, O2 is harmless, H2O2 is there. This has to be taken care. And this is taken care by an enzyme called catalase enzyme. Since its manganese is present, manganese catalase enzyme, since this is also saves the cells from dying. So, it is also oxidative stress reliever, works ROS, reactive oxygen species. And if you look at the catalysis of this one, there is a huge amount of turnover is done by these enzymes, and so as you can see that it converts, and finally it converts to the the water and the O2. So, they both are harmless. Okay, so let us look at the reactive center present in the manganese catalase. Manganese catalase has a slightly different kind of a center than what the manganese superoxide dismutase has got. Manganese superoxide dismutase has got is a monomanganese center, manganese catalase has got dimanganese center. Now, look at the dimanganese center, the protein is shown over here. This is one of the subunits of this, and if you just focus a bit on this side, you can see one manganese ion here, another manganese ion here. This is labeled as manganese 1, labeled as manganese 2, and surrounded by some groups which are directly bonded, some groups which are directly bonded, and that is what you can see here. You can see here one of the histidine is bonded, another glutamic is bonded. One of the histidine is bonded, one of the glutamic is bonded. Also is connecting these two manganese is a bridged glutamic. So, it is a dinuclear, a perfect bridged dinuclear complex. Okay, as you can see that at one stage it has a bridging of this OH as well during the reactivity of this. Okay. So, this you understand the active side, the metal side. We are studying the biological inorganic chemistry or inorganic chemistry of life. So, we are interested at the metal center, and therefore, we are looking at the things happening at the metal center. Now, let me draw your attention to the following aspect of it. Okay, so what is this enzyme? The manganese catalase, and you have the two manganese centers here, and the manganese 2 and manganese 2, and you have the bridging, etc., etc., and this will take up the hydrogen peroxide, and there is a water present, water will be displaced, nothing to worry. So, hydrogen peroxide is forming into this, and removal of this water will lead to the manganese center to be oxidized. You see that the manganese center is oxidized. Manganese center is 2 plus 2 plus 2 will go to the manganese center in the plus 3 plus 3. This B is the one which is the base, which is nothing but a carboxylic group in the protein side chain of the one of the protein moieties. When it takes up the proton, it becomes the BH plus, okay, and that is what it says. So, that is called general base reaction, and that proton is given away, and that will go into the water. So, therefore, you have the and this stage where the manganese 3, dimanganese 3, like in the previous case it was manganese 3 to manganese 2, here it is manganese 2 to manganese 3, it is a reverse kind of a reaction. This manganese 3 now, dimanganese 3 will take up another H2O2 molecule, and this H2O2 molecule is again interacting with this, and that leads to the removal of the O2 moiety out of this, and then leaves the water molecule in this. And you see that there is a simple carboxylate bridge here, but when it comes to the dimanganese 3, it demands O bridge, that is called O2 minus bridge. So, it is the manganese to manganese is manganese 2 to manganese 2, you have only carboxylate bridge. Okay. But when it goes to the manganese 3 to manganese 3, so you have one, that is okay, and this is what we remove is O2 minus. So, this is oxo species. This oxo species comes when we have a manganese 3 kind of thing, and that is what you see in the structure over here. This is a bridging, and this bridging will again fall down, and this goes as a water, as you can see in this, and it will form as a O2 is. So, in the first case, in the first step water is coming out, and the second step O2 is coming out. So, therefore, H2O2 is converted into the water and the O2. You can see the reaction here. H2O2 plus 2 electrons plus 2 protons giving to H2O, H2O2 giving 2H plus O2 plus 2L. The total reaction is 2H2O2 going to water and oxygen, and this is what the thing is, what is happening. Understand that? So, this process is a reduction process, and this process is an oxidation process. So, in the previous case, the first step is oxidation, second step is reduction. With respect to the metal point of view, first step is the reduction, second step is oxidation. Here it is the other way around. So, it goes from the 2 to 3 and 3 to 2, there 3 to 2 and 2 to 3. So, that is the kind of difference. So, these are some extra reactions in presence of the ammonia or ammonium hydroxide. You can try to trap this particular species, and you can take out and add the H2O2, and you can get the bridging one too. So, this part of the cycle is not very important. If you understand these 4 things is mostly good enough. Okay, going dimanganese 2 to dimanganese, the peroxo bridging to the dimanganese 3, and then losing out the water, and the next hydrogen peroxide binding will take back to the manganese 2 and the O2 kind of thing. So, now you understand together when you add the first part is the superoxide is dismuted to the O2 and H2O2. By the catalase, the H2O2 is dismutated to the water and O2. So, therefore, enzymes of manganese SOD plus manganese catalase, so will protect from oxidative stress. Together. So, this is an important aspect that one need to understand in this. Okay, let us go to the next enzyme. The last one in the manganese series is the bacterial photosynthetic apparatus. It is a very huge one, and I will show you on the next slide the reactions, etc. All that we are interested in this is the where, as an inorganic chemist. So, we are interested in the species where this particular region where the oxygen is being evolved, okay, and that is where is shown over there, the water going to O2, and this is where you have the manganese cluster, and this is what comes under the photosystem too. The rest of the things are not important for biological point of view, where what kind of proteins are there, and this is a membrane, etc. So, green plant photosynthesis. Let us look at the kind of reactions that happen, and you know very well in the photosystem there are 2 parts of the reaction. One is the light driven reaction, other is the dark reaction. So, that means there is a light and there is a lapse and dark, there is a light, there is a lapse which is a dark. So, light and dark stimuli. In the light, what happens is that the light is necessary to oxidize the water to the O2. Of course, you are using NADP, and of course, in the process that ATP synthesis takes place. So, the NADP gets reduced and water gets oxidized to O2. And the second part is the dark part, which we are not going to be concerned in this particular biological inorganic chemistry, but just see the reaction. The CO2 is converted to the C6H12O6, which is carbohydrate. Therefore, you have a reduction occurring in this, and so this whole thing is by the reduced. So, NADPH protons, the 2 electron reduced. Okay. So, therefore, you have the 12 ATPs is converted. So, you have a lot of ATP consumed in the second reaction, ATP is synthesized in the first reaction, but however, we are concerned with the first reaction. The same thing is shown over here. There is a light driven reaction and the dark driven reaction. So, the light reaction, the light plus water is converted into oxygen, and then carbon dioxide is converted into the glucose. Okay. So, let us look at how this particular thing is seen in this enzyme. Enzyme has got two parts, one is photosystem 2 and other is photosystem 1. We are not at all involved in explaining the photosystem 1, but we are involved in explaining the photosystem 2, and that too in the photosystem 2 at this stage you have the manganese cluster. We will be looking at this as an important event. And you see there are different reactions happening. I will explain you. First look at this particular thing in form of energy given in the form of electron volts. You can see it also can be converted into the potential. So, if you look at this particular center is the one which is involved in the oxidation of the water. So, when you oxidize the water, obviously you are releasing the electrons, and these electrons are carried over here, and this is the antennae where it can collect the photons, and that will take the excited state, and at this excited state the positive charge and the negatively charged electron will separate, and this separated electron will pass through all of these, and this is called a funnel, and these are different proteins which is not a part of this story now. There are different electron transport proteins. I will be explaining electron transport proteins when I come to the iron story and when I come to the copper story also, and this will go here and this part will go into the photosystem 1. So, let us focus at this particular thing, what happens in this. So, before going to that, just not to frighten you, this is the kind of a photosystem, cyanobacterial photosystem 2, huge protein. We are interested in this particular region where circled, where the tetranuclear manganese cluster is involved. So, that is where the water oxidation happens, and that is what we are going to look at the thing. Now, so I talked to you that when the light impinged on the source and the oxidation of the water occurring, releasing the electrons will go through this kind of a funnel. So, you can see these arrows are showing the direction of these ones, and all these groups collect the light and they are light harvesting. So, you have a coupled light harvesting plus the electron transporting, and beyond this it will go to the photosystem 1. We are not interested in that. Okay. So, that is how the electron transfer. Now, let us see very close to the manganese center. As I told you earlier, it is a manganese tetranuclear cluster. It is a tetranuclear manganese cluster along with calcium. Now, there are 5 ions. You can see here the purple ones is manganese 1, manganese 2, manganese 3 and manganese 4 and calcium. So, manganese 1, 2, 3 and calcium form a kind of a distorted cubane structure where the manganese 4 is protruding outside. So, you can sit leisurely and go through what are the connections with the protein is already shown here. What are the secondary interactions are all shown here. So, these dot lines are shown through the secondary interactions. So, all these species, the manganese is stabilized by the coordinations, as well as the oxo species are stabilized by all the red ones are the oxo species by a lot of secondary interactions which are shown by the dot kind of thing. So, by looking at this you will get. So, the important aspect in this is the role of this manganese cluster. In fact, the manganese 1, 2, 3 is not directly involved in oxidizing the water to O2, but it is the manganese 4 and the calcium, these are involved. But all other manganese are important because there is a lot of electron redox processes happening. So, and there the manganese 1, 2, 3 will play a role. So, you can also see how this particular light harvesting system, and this is where actually the manganese 4 cluster and then water converting into this. So, we are not interested in the rest of the part. As you can see here, the cluster, the cubane and the manganese outside converting. So, therefore, it is this manganese and the calcium which are important in this. Now, look at the kind of steps that we have. As I told you, it is a light and dark and light and dark and light and dark. So, the original state of the enzyme is called S0, and after one pulse of light it is called S1, next pulse of light is called S2, next pulse of light S3, next pulse of light S4, and then go back to this. So, as a result of this, what are you seeing here? Manganese oxidation states are changing. So, what is in each case? One electron is coming out, 1 proton, 1 electron, 1 electron, 1 proton, 1 electron, 1 proton. You count all of these, 4 electrons and 4 protons you can see, and of course, there is a water entry into this, and that is. So, the total reaction is 2 waters converting into O2 plus 4H plus 4 electron, and that is what is done by these ones. But actual water binding and conversion into O2 is done between the calcium and one of the 4th manganese, not with the other things, but all other manganese are also absolutely important. So, 3 of the manganese 3 plus, 1 manganese 4 plus, what it means is, and then one oxidation taken place, therefore 2 are in 3 plus, 2 are in 4 plus, and then one more electron. So, 1 3 plus 3 of the 4 plus, and at this stage either all 4 plus, not very clear, 1 3 plus, 3 of them are 4 plus, but there could be a radical also on the ligand side, we do not know exactly. So, the well understood regions are S0, S1 and S2 in fact, the other states are not. In fact, in the literature if you go and see, a lot of small molecular complexes are already being synthesized and studied very well for S0 system, S1 system, S2 with the tetranuclear manganese complexes not known. But in this course I am not taking up all those details in this. So, we understand that totally all these 4 electrons and 4 protons happening through these oxidation states. So, as I told you, one of the important aspect in this is that all the 4 the manganese ions are not directly involved with the O2 water, but all the manganese ions are important in the electron transfer, as you have seen in the previous the cycle that is shown there. But only one of the manganese and the calcium center are involved in water activation, and this is further supported by one of the anion support. This anion will be favoring this hydrogen bonding system and favors. So, the water binds from the manganese and from the calcium center, and the conversion of this water into the manganese oxo species, and that is where the oxidative state occurs. And this oxidized manganese 4 is reactive, therefore, it reacts with one of the neighboring waters and form a OO bond, which you can call this as a peroxo bond, kind of a equivalent to peroxo bond, and this peroxo bond is further activated by breaking up and to give the O2. So, there are other kinds of mechanisms in the literature we are not taking up. This does not mean other mechanisms do not occur. Why other mechanisms are not there? Because we do not know, as I told you in the previous slide, the S3 state is not well understood, and S4 state of course is not understood, because this goes very very fast. S0 is very well known, S1 is very well known, S2 is very well known. Therefore, there could be more than one different kind of a mechanism. That is the reason why we are saying all this. So, though there are 4 manganese, it is only one manganese ion which is directly involved in binding to oxygen, in converting the binding to water and converting into peroxide, and then to O2. Okay. All other manganese are important for in the electron transfer funnel in this thing. So, overall what we have seen? Overall what we have seen in the manganese story, manganese has large number of oxidation states, but we see that the important things are 2 plus, 3 plus, 4 plus, very rarely maybe 5 plus, not so much so. 5 plus and beyond are not too oxidative in nature, and below 2 plus are too reducive in nature, therefore, these are not. There are variety of oxidative, redoxative kind of enzymes are there, but I have tried to give you only a few of those examples, and the ribonucleotide reductase, the superoxide dismutase, the catalase, the photosystem and dioxygenase, of which 3 cases I have explained the mechanism, that is superoxide dismutase, which converts the superoxide into the hydrogen peroxide plus O2, and this is further taken up by this hydrogen peroxide is further taken up by catalase, and catalase is a dinuclear manganese system, and this will convert the H2O2 into O2 and plus H2O. So, overall SOD and catalase together will save the cells, will save the tissue, will save the body from the oxidative stress or reactive oxygen species, ROS. So, therefore, it is important, I have explained that. The mechanism in one case is going from 3 to 3 oxidation 3 plus to 2 plus in the other and reversal, in the other case 2 plus to 3 plus and the reversal. The one is mononuclear, other is dinuclear. Then the other enzyme that I have explained to you is the not an enzyme, a part of the enzyme in the photosystem 2 where the tetranuclear manganese cluster is involved. Though the tetranuclear manganese cluster is involved, it is associated with another calcium, and calcium plus 3 manganese will form a distorted cubane structure, and the fourth oxygen which is standing outside or sitting outside is one which is important in direct binding to water, converting to manganese oxo species, converting this into the O2 peroxo species, then further converting into O2. But all the other 3 manganese are also important. As you can see, the manganese are going through the 3 plus to 4 plus and etc. that we have seen through S0 to S1 to S2 to S3 to S4 back to S0, and I told you S1, S0, S1, S2 are very well understood. S3 and S4 is not much understood. S1, S0, S1, S2 cases, there are a large number of model complexes are built, and I think we will probably stop the story of manganese at this stage after explaining these 3 enzyme mechanisms. Then we will go into the next class in the iron enzymes. Thank you very much.